This video is part 2 of DNA replication, where we will get into the details of DNA replication. When you look at this picture down here, you'll see the original DNA. Here's the top strand of the original DNA, 5' to 3'. And here's the bottom strand of DNA. Okay, so if this is 5' to 3', this is 5' to 3', then this side will be 3'. Here's the original DNA to 5'. When DNA replicates, the first thing it does is open up a section of the DNA. This open area on the DNA is called the replication bubble, or the origin of replication. DNA replication actually occurs at both sides of the replication bubble, and these two sides are called replication forks. Today, we'll just be focusing on one side of this replication bubble. We're going to look at DNA replication at one replication fork, this one over here. So just to avoid confusion, I've drawn a little line to indicate that we're talking about this side of the replication bubble. So right here we have a replication fork. The purpose of this video is to go through the process of DNA replication and to highlight the different enzymes that are involved in DNA replication. If you use different colors, I'll be color coding this video. I'll also be using shapes to denote the different types of enzymes. So the first enzyme we're going to address is the enzyme that is responsible for opening up the DNA. I'll draw that as a triangle. In real life, it doesn't look like a triangle, but I'm just trying to make my point. So the enzyme that opens up the DNA is called helicase. Helicase unwinds and unzips the DNA to break the hydrogen bonds so that you can see the complementary nucleotides on this strand of DNA and the complementary nucleotides on this strand of DNA. So we can go ahead and add those complementary nucleotides in. A would pair with T, C with G, G with C. It gets harder as the nucleotides get farther apart, right? So go ahead and add your complementary nucleotides as well. Okay, so now we can see both sides of the DNA. This original DNA will act as a template for the new DNA being synthesized, and the helicase will hold that DNA apart. As the DNA is pulled apart, the twists in the DNA get tighter and tighter. So to prevent the DNA from snapping, we have another enzyme called topoisomerase that cuts and rejoins the DNA to relieve that pressure of the DNA, of the twisting of the DNA. Okay, so it relieves the twisting stress in the DNA. Also, since these two DNA strands are single-stranded right now, and DNA isn't as stable when it's single-stranded, the cell puts some proteins on the outside of the single-stranded sections of the DNA to stabilize it. Those are called single-stranded binding proteins. So these three proteins will work together to open up and stabilize the DNA so the DNA replication can occur. And right down here, we're going to just kind of summarize what these three steps do. Okay, or these three enzymes do, sorry. So helicase unwinds and unzips the DNA. It breaks the hydrogen bonds between the nucleotides. Next, we have this topoisomerase right here. And topoisomerase is responsible for relieving the twisting stress. By breaking and rejoining it. The last enzyme in this first category are the single-stranded binding proteins. The job of these proteins is to stabilize the open DNA strands. DNA replication has a couple of problems that it has to overcome. The first problem is the primary enzyme that helps new nucleotides join to make our new DNA strand is called DNA polymerase. But DNA polymerase is a fussy little enzyme, and it won't join to the DNA unless it has a double-stranded area to join to. So let's look up here. We have our original DNA, and we want to use it to synthesize new DNA, but this is a single-stranded area. Luckily, we have another enzyme that's less fussy and is more willing to get in there and help us make a new DNA molecule. That enzyme's name is primase. So primase joins onto the original DNA strand, and it moves down the DNA, and it takes nucleotides that are floating around in the nucleoplasm, which is like the cytoplasm inside the nucleus, and joins them to the DNA. However, this enzyme primase doesn't add DNA nucleotides, it adds RNA nucleotides to create something called an RNA primer. So here's my RNA primer. You can tell it's an RNA primer because instead of using T, it uses uracil as a nucleotide. Okay, so here's my RNA primer. Thank you, primase. Now we have a double-stranded area. Yes, it's a DNA-RNA hybrid, but it is a double-stranded area. So our next enzyme can come in to help out. That next enzyme that's going to come in is called DNA polymerase. The model that we're studying is actually a prokaryotic, meaning bacterial model. So this enzyme is called DNA polymerase 3, but a similar DNA polymerase enzyme is used in human DNA replication as well. So DNA polymerase 3 is going to move down this DNA, and it's going to add new nucleotides. Remember, new nucleotides can only be added on the 3' end. So this new DNA is going to be made in the 5' to 3' direction here. All right, so we have now we're adding DNA nucleotides, etc. So this is our new DNA that's being added using the original DNA as a template strand, and it's being added in the 5' to 3' direction as new nucleotides are added on the 3' end. Again, these nucleotides are just floating around in the cytoplasm. So there's our new DNA right here. Our cell, however, does not like the fact that we have this DNA-RNA hybrid. So it gets another type of enzyme involved. That other type of enzyme is, I'm going to draw it like a pair of scissors. Well, I can't draw that well, but you get the idea. This other type of enzyme is called DNA polymerase 1. This DNA polymerase 1 is going to cut out the RNA nucleotides and replace them with DNA nucleotides. So now we have a piece of DNA here and a piece of DNA here. The bonds on the side, the phosphodiester covalent bonds on the side, are made as the nucleotides join together. So we have a fragment of DNA here, and we have a fragment of DNA here. But they're missing a covalent bond between these two fragments. Luckily, we have another enzyme. So once DNA polymerase 1 cuts out the RNA primer and adds the DNA in there, we have another enzyme called ligase, which is going to come in and help that create that covalent bond between these two fragments. Okay, and that's going to allow us to have a continuous DNA strand here. So let's write down what all these different enzymes do that we just drew a picture of. So first, we have our primase. Sometimes it's called RNA primase. Primase is going to add RNA nucleotides to the DNA. So that DNA polymerase 3 has a double-stranded area to attach. Okay. Next, we'll have DNA polymerase 3 coming along. DNA polymerase 3 is going to read the original DNA. in the 3' to 5' direction, and it's going to synthesize a new DNA strand in the 5' to 3' direction. Okay. To make the complementary strand. Okay. Now again, we had that crazy RNA hanging out in there, RNA primer that helped us get started. But our DNA polymerase I, our little scissors are going to come along. They're going to cut out the RNA and replace it with DNA. And finally, we'll have our ligase. Our ligase is going to form covalent bonds between the DNA fragments. It's going to kind of act as the glue there. So I want to look at these enzymes and kind of write a list of steps of how DNA replicates. And in that, I want to talk about the leading and the lagging strand. So we're going to write our steps over here. I'm going to give you 5 steps for DNA replication. So in step 1, the helicase unzips the DNA at the replication fork. And of course, the single-stranded binding protein stabilize it, and the topoisomerase relieves the twisting stress. And next we have the primase. The primase creates an RNA primer, which makes a DNA RNA double-stranded area for DNA polymerase III to attach to. Okay. Next, the DNA polymerase gets nucleotides, 3 gets nucleotides from the cell, reads the original DNA template, and synthesizes a new DNA strand in the 5' to 3' direction. And the original DNA is the template here. Now I want to talk briefly about the leading and lagging strand. So DNA is always replicated in the 5' to 3' direction. This top strand, once you get the DNA polymerase to bind, as the DNA opens up, this top strand can be synthesized continuously towards the replication fork, and it just keeps being synthesized as the DNA opens up. So this is known as the leading strand. Because it continuously replicates towards the replication fork. The lagging strand is a little different. Same thing happens, right? Except you can't see all this DNA at first. The DNA hasn't opened up that far. So let's say the DNA has only opened up this far. So you have your little RNA primer here. Then you make your new DNA here. Then again, DNA polymerase I comes and cuts out the primer. You make your DNA. Okay. But again, you have to add another primer, another piece of new DNA, and we're replicating away from the replication fork as this DNA opens up so we can see this piece. Again, DNA polymerase will replace it with DNA. So you end up having a discontinuous replication, you end up having all these fragments of DNA that eventually are all glued together by ligase to make a continuous strand of DNA. But this replication process is jerky. One fragment is replicated at a time. These fragments, by the way, are called Okazaki fragments. But because this DNA replication process is jerky and a little piece happens like at a time, so we put the primer on, add the new piece of DNA, then the DNA opens up more, we add a primer, a new piece of DNA, replication occurs away from the replication fork. This is called a lagging strand because it is discontinuous or synthesized in segments, so it takes a little bit longer. So we should add that to step 3 over here. Okay. Both occur in the 5' to 3' direction, right? So both leading and lagging occur 5' to 3', but leading is continuous and towards the replication fork. Okay, whereas lagging, again, DNA replication always occurs 5' to 3' because of how the nucleoside triphosphates work. But the lagging is discontinuous because it has to occur in chunks away from the replication fork. Okay, so after the DNA segments are replicated, okay, DNA polymerase 1 cuts out the RNA and replaces with DNA. And then ligase has to act on both the leading and lagging strand. It glues 2 fragments together on the leading strand and lots of fragments together on the lagging strand. But ultimately, you'll have 2 copies of identical DNA using this process. In a prokaryote, you have one origin of replication, one replication bubble, and replication just occurs in the circular chromosome until it's done. In eukaryotic DNA replication, you have many replication bubbles, and DNA replication is occurring in many places on our chromosomes at the same time. Because of the fact that our chromosomes are linear, we have some lost DNA at the end each time we replicate, but that will be in a different video.